We have discussed nephron in detail, mainly the two parts, the glomerulus and the proximal convoluted tubules. A nephron, basically nephron is composed of two parts, the renal corpuscle and renal tubules. Renal corpuscle is, for example, this is nephron. This is nephron. This is proximal convoluted tubules. This is loop of henle. This is distal convoluted tubules, and it is draining in collecting ducts. Right? The first part is, for example, this is the arcade artery or the interlobular artery, and this is the afferent arteriole. This is the glomerulus, and this is the efferent artery. Right? The glomerulus, this is glomerulus and Bowman's capsule. This part is called renal corpuscle. This is basically afferent arteriole, and this is efferent arteriole. Right? This we have discussed yesterday. This part, the proximal convoluted tubules. We have discussed the absorption and reabsorption processes in what is reabsorption? Reabsorption is the transport of molecules from tubules into blood. And what is secretion? The transport of materials from the blood into tubules. We have discussed this in detail. Second, this area is called loop of henle. In loop of henle, this part is called the descending limb of loop of henle. Right? And this red part is called ascending limb of loop of henle. Right? Why it is separated? Because the channels, the physiological functions here and the ascending limb they are different. They are different from each other. One difference is this descending limb is highly permeable to water, and this is impermeable to solutes. And this red area is highly permeable to solutes, and it is impermeable to water. Right? So we will discuss this. But according to the loop of henle, nephrons have two types. One type of nephrons have short loop of henle, and one type of nephron have long loop of henle. For example, I am taking the kidney here. Generally, for example, this is the kidney, and kidney have two parts. One is the renal cortex, and the other is renal medulla. And renal medulla, we have a special type of structures called pyramids. They are called pyramids. For example, these are pyramids. And here we have one type of nephrons called the cortical nephrons. For example, these are cortical nephrons. In cortical nephrons, we have proximal convoluted tubules and short loop of henle and collecting duct and distal convoluted tubules. The second type of nephron is the juxtamedullary nephron. They have the glomerulus near to the corticomedullary junction and having very short proximal convoluted tubules and very long, loop of Henle very short, distal convoluted tubules and collecting ducts. Right here, I have discussed that in medulla there is a lot of salts, a lot of solute concentration. Right. In cortical nephrons we have peritubular capillaries, right? Peritubular capillaries, and in juxtamedullary nephrons we have vasa recta. Vasa recta is a long tube of after the glomerulus, and they are covering the loop of Henle. What mechanism will occurs in the vasa recta we will discuss in detail. They are called the counter current exchanger mechanism, right? And it is used for the dilution of and concentration of urine, right? So we will discuss this loop of Henle in detail now, but before going to physiology, this loop of Henle have also cuboidal epithelium. What is the difference between the epithelium of the proximal convoluted tubules and the loop of Henle epithelium? The main difference is, for example, here we have, this is Bowman's capsule, this is proximal convoluted tubules, for example, and I have told you that it have a brush borders like this. They have brush borders on lumen side of nephron for the increasing the surface area of the cells, right? But for example, this is loop of Henle, this blue is, this is loop of Henle. It have also cuboidal epithelium, but they have no brush borders on inside of nephron. They are simple cuboidal epithelium, right? And where there is epithelium, there will be the exchange of materials, right? Because this is also composed of epithelium, and the blood vessels which are following this renal tubules is also composed of simple squamous epithelium. So the exchange and the reabsorption, the secretions occur very easily in these areas, right? So now we will focus on this loop of Henle, but just recap the mechanisms in proximal convoluted tubules. We will take one cell from proximal convoluted tubule quickly. For example, this is Bowman's capsule. This is Bowman's capsule, and this is proximal convoluted tubule. Proximal convoluted tubule. And in proximal convoluted tubules, we have the cell having brush borders. This is, for example, the cell right, and this area is called tight junction. It is called tight junction. The paracellular transport occur in between these two cells, right? First, this is the glomerulus. The afferent and efferent arteriole. The osmolality of the blood is 300 milliosmol per liter, right? And under this what is osmolality? Anyone? What is osmolality? The content of solutes in per liter or per ml of water. This is called osmolality. It's mean that when the solutes are increases, the osmolality increases, and when the solutes concentration is decreasing, osmolality also decreasing. So for example, I am taking this one pyramid from the kidney. This is, for example, one pyramid, right? And this is the cortical or cortex area of kidney. So for example, here I have glomerulus, and this glomerulus is placed in a cup-like structure called nephron. This is very important. This concept is very important for your MCQs, right? For example, this is the proximal convoluted tubules, and this loop of Henle is dipping in the pyramid or in the medulla of kidney, and this is, for example, distal convoluted tubules, and this is collecting duct, right? I have already told you that in pyramid there is a lot of solute concentration. It means that the osmolality of the pyramid is greater as compared to the cortex. The blood plasma osmolality is 300 milliosmol per liter. Here this area is, for example, this is proximal convoluted tubule. Here also the osmolality is 300 milliosmol per liter. It means that the solution here is isotonic. Do you know isotonic, hypertonic and hypotonic solutions? When the plasma solute concentration and the solute concentration in other solution is same, it is called isotonic. When the solute concentration in the solution is higher than blood plasma, it is called hypertonic. And when the solute concentration is low, it is called hypotonic. Here are three concepts of osmolality. Three concepts of osmolality. Right? When the solute concentration is high, what are the solutes mainly? The sodium and chloride. The sodium and chloride. For example, high sodium and high chloride ion. The water concentration will be low. This type, it means that the osmolality is high, and this type of solution is called hypertonic, or it is called hyperosmotic solution. This first type. How we can clarify this? For example, this is the blood. This is the blood, and this is interstitial solution. If the solute concentration in blood is low as compared to this solution, if the water concentration in the blood is high as compared to this solution, it means that the osmolality is low in blood and osmolality is high in the solution. This solution is called hyperosmotic solution, right? When, for example, the concentration of sodium is low, the concentration of chloride is low, the concentration of potassium, magnesium is low, all the solutes, and the concentration of water is high, it means that the osmolality is low, and this type of solution is called hypotonic, or it is called hypoosmotic solution, right? These concepts will be used in the loop of Henle, right? And the next one is, for example, the sodium concentration is equal to the sodium concentration in blood, right? The water concentration in solution is equal to the water concentration in blood. It means that osmolality of water is equal to osmolality of blood. This type of solution is called isotonic solution. For example, if we are injecting something into the veins directly, all the injections, they are isotonic solutions. Isotonic, the pH, the osmolality, they all are same to blood. There are three types of solution: the hypertonic solution, the hypotonic solution, and isotonic solution. Or hyperosmotic solution, hypoosmotic solution, and same osmotic solutions are isotonic solution. So here the concentration of salts NaCl, it is very high in the medulla of kidney, right? So when the concentration of solutes is increasing, it means the osmolality of this area is increasing. For example, here the osmolality is 500 milliosmol per liter. For example, here it is 700 milliosmol per liter. For example, here it is 900 milliosmol per liter, and here it is 1200 milliosmol per liter, right? So when we are comparing the osmolalities of the cortex and the medulla is getting more osmolality because it taking the solutes from the urine, also from blood, right? We will discuss this concept, but this is very important. Remember this, that in medulla the osmolality is increasing when you are going downward, and when you are going upward in ascending limb, the osmolality of this area is decreasing. For example, here it is 1200. For example, you are going downward and descending limb, the osmolalities are increasing, but when you are going upward, the osmolalities are decreasing, right? And it becomes same with blood in last, right? So this is the concept about the osmolality, right? Now what is the mechanism in proximal convoluted tubules? Just recap quickly. We have sodium, potassium, ATPase. Remember this, it taking 3 sodium from the cell outside and it taking 2 potassium into the cell. For example, sodium is leaving and potassium is getting in, and it cause the sodium concentration gradient here, right? And we have a special transporters here called sodium glucose co-transporter, and it takes sodium as well as glucose into this cell, and this transport is called secondary active transport, and this is called primary. For example, primary, first is primary active transport, second is secondary active transport. Amino acids and lactic acid, they are also following the secondary active transport. What is the next thing? The next thing is the calcium, magnesium and potassium at following paracellular transport, right? Lipids and urea, they are following transcellular transport. Water and sodium, they are also reabsorbed from these areas, and this type of transport is called obligatory water reabsorption. When the water is following a solute or solute is following the water, it is called obligatory pathway. So this is obligatory water reabsorption. These are the mechanisms which occurs in proximal convoluted tubules. How many sodium is reabsorbed in proximal convoluted tubules? Proximal convoluted tubules. First, sodium. How many? 65%. Water, 65%. Glucose, 100%. Glucose, if some of the glucose are remaining in the lumen, this cell has the ability to take these minor quantities of glucose from the lumen. If you are normal, if the kidney is normal in physiological activities, but if the kidney have some type of issues, it will remain this glucose in the urine, and when you trace the glucose in urine, it means that you have some dysfunction in your kidneys. Amino acids, 100%. Lipids, 100%. Chloride ions 50 to 55%. Potassium ions 50 to 55%. Right? And magnesium bicarbonates, they are also reabsorbing from this area. One mechanism is the bicarbonate ion. Bicarbonate ion is not directly reabsorbed into the blood. They are using the carbonic anhydrase. How? The water and carbon dioxide. Water and carbon dioxide, they react and forms H2CO3, the carbonic acid. This thing is very unstable and break into hydrogen ion and HCO3 minus. Right here, this hydrogen ion here react with HCO3 minus and form H2CO3, and it break down into bicarbonic anhydrase CO2 and H2O. Right? And this bicarbonate ion reabsorbed into the blood for maintaining the pH of blood. This is the mechanism. The next mechanism is of insulin, the small proteins, the endocytosis. By endocytosis, the insulin is reabsorbed from into the cells, and in the cells it is breakdown. It is breakdown by lysosome, and after breaking down, it is reabsorbed into the blood. This was the mechanism of proximal convoluted tubules. Now we will discuss the loop of Henle under the mechanisms of this proximal convoluted tubule, and we will discuss one very important mechanism. Loop of Henle is very simple, right? There are no a lot of mechanisms in loop of Henle. There are only two mechanisms. One is the counter current multiplier mechanism. One is counter current exchanger mechanism, and one transporter is working, which is called the sodium potassium 2 chloride co-transporter. Right? So let's